UiTM Office of Deputy Vice-Chancellor, Research and Innovation for this invitation to stay motivated weekly sharing session with 5-star UiTM RG and RIG webinar series 4.0 2022. And of course, I would like to thank all of who are here, all who joined today, that come and give your time to this session. I wish that everyone here, inshallah, will benefit and enjoy this session till the end, inshallah. All right, I'm basically Dr. Nik Raikhan Nik Him from School of Chemical Engineering, College of Engineering. I am the moderator for today's session, and our speaker is Associate Professor Dr. Fazlena Hamzah from Biocatalyst and Biobased Material Technology Research Group. And she's also from School of Chemical Engineering, College of Engineering, UiTM, Shah Alam. So the topic will be Biocatalyst in Product Development. So I guess I'm not going to take longer time. So I think we go straight to the show. Let us hear from our Associate Professor, Dr. Fazlena Hamzah. Please, Doctor, you can start.
Thank you very much, Dr. Nik. Let me share my slide first. Right, so boleh nampak ke slide saya?
We can see.
Alright, sorry. Okay. Alright, okay. Assalamualaikum warahmatullahi wabarakatuh and very good morning to everyone, everybody. So I'm Fazlena. So I'm from Biocatalysis and Biobased Material Technology Research Group, BBMT, what we call it BBMT. So the location is at School of Chemical Engineering, College of Engineering, Universiti Teknologi Shah Alam. So generally our research group, our team members actually focuses on the utilizing of the catalyst in a product development. So for today, I would like to share some of the activity and the research works that has been done within our team members. Okay. All right, so before we go further, so what motivates us to do this research? Mostly because of the biocatalysis itself. Actually, we used to accelerate the process, to speed up the reactions process due to the very interesting property of this catalyst, which is very specific to the substrate, selected substrates, effective biological catalyst, and it's able to lower the activation energy so that the process can be done faster. So when we talk about biocatalyst for our group, we focus on the enzymes, which to convert certain organic product into the value-added product. Okay, so in this study, we look into the global scenario actually, especially in terms of the chemical industry and the associated industry that tends to produce complex product, but with want to stay in the green approach, sustainable and environmental friendly due to demands in the community, especially in terms of the food industry, pharmaceutical, agriculture, fine chemical, and so on. So when we look into this scenario and this potential, we look into the possibility to expand the potential of the enzyme itself in industrial biocatalyst. Right, so what we can summarize that biocatalysts offer beneficial activity, beneficial reactions as compared to the normal reactions using chemical catalysts. First, in terms of the environmental friendly. So enzyme itself actually environmental component, so it can degrade it in the environments easily. Second is when we use the enzymes, mostly the reaction is occur at mild conditions, almost at room temperature. So it will minimize the side product and minimize the side reaction, so that almost no side product actually. All right. And the third one is ability to be reused or recycle against the enzyme when we use with the under condition immobilized approach. All right. The fourth one is in terms of the selectivity toward their substrate. So that when it's very selective to the substrate means that no side product can be produced during the reactions. All right. So this is initially what made us together in our team to come out with the fundamental or research related with the catalyst, which is enzyme. Okay, so let me introduce our team members. So myself, more focused on the bioprocess. So actually in this research group, we have the combination from the chemical engineering and also faculty of the plantation and agro-technology. So me, Dr. Sharina Fairuz, Dr. Mira focusing actually our specialization in bioprocess, while Dr. Harumi Wini is more towards biodiesel, Dr. Muhammad Zaki Zain Abidin more towards flow insurance, and Dr. Zuraida towards natural product chemistry. And also we have associate member Dr. Nik Raikhan focusing on the biosensor. Okay. So, alright. So our focus area in this BBMT is more towards developments of the biomaterial, enzyme technology, more toward immobilizations and process optimizations. We do on the biofuel, bioproduct, and also biofolins. Okay, let's go detail on the current project that we have in our group. So first, in terms of the developments of the calcium carbonate. So actually for these things, the motivation is come out with the, it start with the compositions, we start with the statistic on the CO2, amount of the CO2 keep increasing in the environment. So we bring the problems into our labs by making the or the industrial conditions. So we try to utilize the CO2 with the help of the carbonate anhydrase enzyme to accelerate the process so that this CO2 has been converted into the calcium carbonate. So this calcium carbonate can be used in the different application, for example, in the pharmaceutical, food industry, and so on. So on top of that, instead of we are using the enzyme, we also doing some enzyme engineering, focusing on the developing suitable support material that can attach enzyme into the surface of the solid material, so that when we use this enzyme, we can recycle or reuse it again for the next processing. So currently, we are focusing on the lapis in Candida artatica with its color, so that the raw material, the support used in this study, we obtain polysonic material that is abundantly in the Malaysia from the palm oil industry. Okay, so the next sekejap tak boleh nak turun sekejap.
It's okay Dr. Faz, take your time.
Got something, saya end slide sekejap
Okay, no problem.
Boleh nampak ke?
Nampak.
Okay, so the second project is handled by, leads by Dr. Harumi. So Dr. Harumi utilizing of immobilized enzymes in productions of the biodiesel. So currently, Dr. Harumi has study different types of the substrate, which is used cooking oil, rubber seeds oil, crude palm oil to determine the best, the optimum biodiesel that can be produced during trans-aspiritation process. Okay, the next one is Dr. Muhammad Zaki Zain Abidin. So Dr. Muhammad Zaki actually doing the research related to the biofolin. So this project has been conducted at Ocean Thermal Energy Conversion OTEX at Teluk Kemang PD. Dr. Sharina Fairuz more towards biochemical synthesis using fermentations. The focus on the xylitol from the different substrate. So then Dr. Mira Dato focus on the anti-obesity, which is focusing on the lipase inhibitor. So Dr. Mira Dato working with the extractions of the active ingredients from the plant's material. So far, she are focusing on the Equalaria malacensis and Equalaria subintegral leaf. Okay, the last one is Dr. Wan Zuraida. So Dr. Wan Zuraida is focusing on the phytochemistry and biological activity from the plant waste material. So currently, she doing the development of the pesticide from the this plant's active ingredients. So this is an example of the antimicrobial activity that has been conducted. So this is our RIG milestone. So actually, macam mana nak cakap? Actually, we still we can consider our RIG still at orang kata fasa bukan advance lagi lah as an initial fasa. So if I can say this is the best practice, it's not the best practice actually, but knowing our team members with our capacity still lagi young lecturer, so we try to be consistent. We try to be consistent start from the RIG established until now. So initially we start the RIG around 2015. So this time actually is more towards determines what is our niche area, what is our interest research activity or research area. All right. So then in 2016, we identified that, oh, okay, this time we need to have grant so that the research can be continuously. And of course, once you have the grant, you need to determine passionate postgraduate. So for 2016 and 2017 is focusing on the securing the grant and the postgraduate student. And that in the same time, since that time, our team member mostly young lecturer, tak ada PhD lagi. So we try also to focus on the career developments of the staff, which niche area she want to focus for PhD. Okay, so then in 2018, this thing we tried to expand a little bit our activity by industrial engagements. So for this time, we're not focusing securing the grant industrial grant first, but more towards collaborations. We, in terms of the raw material analysis, and also to put some orang kata reputations kepada industry, so that they can come to us for the next project and so on. So in 2019, we focusing on the on top of that, we focusing on the publications. So then during 2019, we tried to organize very simple conference within our faculty. So that's that time we organized ISGAT 2019, so with the aims to come out to encourage publications within our team members and also other researcher within faculty. Okay, on 2020, Alhamdulillah, yang ni yang dapat 5 star ni lah. So from the orang kata sikit-sikit lah. So ada achievements within 2020 kat sini. So 2020 ni actually we focus on the international engagement, especially securing the grants and also publications. So publications, joint publication together with the collaborator. Alright, so 2021 we just maintain our activity, and then 2022 now we are focusing on the registrations harta intellect. So whatever research we have, now we focusing on the try to register copyright, patent and so on. This the activity for 2022. Okay, so alright, so this is some of the activity that has been conducted within our RIG. So this is get on top of that, we also conducting workshop to the industry for the beginner lah. It's not advanced yet, but this one for the beginner. The participant is from the industry, other IPT and also the students. And for the school break holiday, we also organize some simple activity with the sekolah budak sekolah rendah lah. Okay, this is on top of that kita pergi pitching, conference and so on lah. So this is our RIG activity. So this is some of the achievements daripada within our team members, IID competition and so on lah. So this is our research collaborator in terms of the funds and then publications we have from the UPM, USM, University of Aberdeen, UIA, then also from the Indonesia too, University for Indonesia. So this is orang kata simple activity we have conducted within our RIG. So that is the activity is consistent during orang kata throughout the years lah, macam tu. So itu je daripada saya punya RIG, very simple. So if you guys have any question, you can ask. Or other researcher want to collaborate with us, you are welcome to our RIG for the discussion and so on lah. Okay, thank you very much. Thank you, Dr. Nik.
Thank you, Dr. Faz. Thank you so much for, to me, a humble explanation, humble, get to know, but very, to me, motivated. Because, yes, of course, because I'm in it sounds like I'm talking good about my own thing. No, but the things that I can see, the way they put the commitment, they from no PhD and then become into having PhD, have specialization on their own PhDs, and they are very good. So I'm going to open this floor to the question. If anyone at all having anything to ask Dr. Faz, or anyone with any idea to support or to make it, happy things today, inshallah. Anyone? All right. Anyway, there's someone texted me. I don't think so. I think that she cannot get through. So the question for Dr. Faz is that, do you produce enzyme and do you sell it? Will you sell it at cheaper price compared to what the industry can give us?
All right. Okay. Generally for during this time lah, we are not producing any enzyme yet. We are purchasing actually from the standard enzyme Sigma and so on. Previously, we do make an attempt to produce amylase. Unfortunately, with the limitations of the apparatus and most downstreams units, so we are unable to obtain pure enzyme itself. So that's why it's very tedious works because our limitation is equipment. So then right now we only focusing utilizing the standard, the commercial ones. But we only focusing on the we purchase the free cell, and that's at the our RIG. We doing enzyme engineering, focusing on the enzyme immobilization and process optimizations only.
Do you have plan to somehow produce?
Kita kalau dapat duit grant yang besar, insyaAllah.
InsyaAllah dan
we can collaborate lah.
Yes, insyaAllah. InsyaAllah dia jadi I pula yang cakap, I juga yang tanya. Okay, let's hear from the floor, please. Someone, some might interested in something at least that you can, somehow giving some ideas, supports from any professors here. Anyone? Because I see, when Dr. Faz was telling about having conference, small conference or small workshop using the RIG, that's an amazing thing. Because to me, personally, commonly, we always see conferences will come from faculty, schools, or college. And RIG with only small limited number, 5-6 persons to run a workshop. Conference is something very valuable and something to learn. So that's one motivation for me and for us, hopefully, somehow. So still any question? Do we want to end this? Like no question? What about you, Dr. Faz? Anything you want to say? And anyone at all?
So what I did. It's okay. Like previously you mentioned that about conference. Yes, actually it can be conducted if you have a very passionate team member. So what can I say that our team member is really committed. So they are eager to do something. If I put something, is it okay to do this thing? Yes, we can do it.
They
So
will say yes.
that is the
That's
positive
one thing
vibe
about them.
that
Yes, very
that
much. I can
encourage
see that.
our movements lah.
And they do it without hesitation. That's one thing. That's what we saw.
Yes.
I'm so sorry, guys. All the floor members who join here. It sounds like I'm putting too positive. Yes, it is a positive thing that I say I can put to this RIG. Well, today is about this RIG, the weekly session. So I'm saying everything positive because it's not only positive about the team, but also positive for us in the UiTM as a whole, as if that all of us are in actually at least one RIG. And of course, maybe we have second RIGs as a partner member, but we are there. So we have to have motivation. We have to move in the best way, best mood, inshallah. All right, I think I don't know how do I do this if there's no question. I cannot keep asking question through the internal, like internal question. Last one, last. Do you have any question, Flo? Anyone? Do you? Anyone? If not, then I take it as everybody got the slides and introduction and everything that Dr. Faz has already introduced to us. One thing. Number two is that the content go absolutely understandable to everyone and it goes smooth and reach everyone. And hopefully after this session, we can have somehow integration of commitment or maybe, some sort of work or mutual research or something that we can bring, that we can understand, something like that. So I think that's all. Is that okay? That's all because it's 10:22. It's kind of, any comment, anything at all? I don't see any comment. So do we agree that we end here or how is it? It's going to happen? What about Nexus? Any comment from Nexus team? No comment? So we can end this? Now I'm laughing. I think, okay, no comment. All right, thank you so much, PM Dr. Nuraza. That is Prof. Nuraza, is it? Like, because I couldn't get to read everything. All right, that's all for today. I think thank you so much for those who came in and thank you so much for Dr. Faz. Thank you so much for Nexus and all the best for everyone throughout the next coming sessions, of course. All right, that's all I think. I'm going to end this session. We can leave. Thank you so much again. Assalamualaikum warahmatullahi wabarakatuh. Bye.
Thank you. Selamat Hari Raya.
Selamat Hari Raya, yes. So we can leave. We have the attendance, guys, in the chat box. So please don't forget to fill in the form.